I am Dr. Sanooj Soni. I am a clinical senior lecturer in critical and perioperative care at Imperial College London and a consultant in anaesthesia and intensive care medicine. We work with predominantly patients who are critically unwell, also those patients who are undergoing major surgery. What brings me to work every day is really to try and make a meaningful difference, not only at the bedside clinically, but also in the lab doing research, which could fundamentally change how we treat critical illness, but also patients undergoing major surgery. If our work leads to a change in clinical practice or a new intervention that improves patient outcomes, that's really the ultimate measure of success and why we do what we do. So the focus of my research is really trying to understand how tiny particles called extracellular vesicles are released from cells during stress or injury, because they are like biological postmen and effectively molecular messengers that travel through various bodily fluids, influencing how target cells and then the organs can respond to both injury and infection. By targeting extracellular vesicles, we are developing strategies to try and reduce complications after surgery and during critical illness. And this could mean faster recovery through long-term disabilities, and most importantly, it could end up saving lives, which is why we do this research. Our analysis of extracellular vesicles was very time-consuming and technically challenging. Traditional Western blotting lacked the sensitivity and reproducibility that we desperately needed, especially when working with particularly small volumes of samples from critically unwell patients or patients undergoing major surgery. The most common question or comment about the Jess from colleagues is really how do we manage to get such clean and consistent results with such small patient samples. And the Jess really tends to impress people when they see the data output compared to traditional Western blotting. Critical feature is the automation and reproducibility that Jess offers. As I'm someone who's overseeing multiple projects, being confident in the consistency of our data whilst reducing hands-on time is invaluable. It allows our researchers to generate high-quality data very quickly without spending hours on complex protocols, which is a huge boost to our productivity. With the Jess, we've been able to expand our scope of research significantly. We're now investing in more complex signaling pathways and protein markers that previously were beyond our reach due to technical limitations. And this opens up really whole new avenues and possibilities for biomarker discovery and therapeutic signaling. So I think that Bio-Techne tools are having quite a significant impact on life science research, and they're able to help accelerate discoveries by making complex analysis more accessible and reliable. And tools like the Jess actually bridge the gap between discovery and clinical application, which is especially important in fields like critical care research. I think the factors that are most important to us are the responsiveness and scientific partnership, which are pretty much key. We need companies that don't just sell us equipment, but actually spend time supporting us in troubleshooting and optimizing our experimental protocols, particularly when we're doing quite tricky work with extracellular vesicles. And Bio-Techne has really provided excellent support and understands the real challenges that we have with translational research.